Hi, everyone. Welcome to the single cell analysis session. So in this session, we'll have 4 talks. If you have questions for the speaker, please type it in the chat box. And then we will answer the questions during the Q&A session. So if we cannot get to your question during the Q&A session, you can email the speaker about your questions, or you can use the Cafe Connect platform through the conference website, where you can network with others. Okay, so our first speaker today is Benjamin Chidester, and he will talk about SpiceMix integrative single-cell spatial modeling for inferring cell identity.
Hello, everyone. My name is Ben Chidester, and today I'll be presenting our new tool called SpiceMix, which is a method for integrative spatial modeling of the spatial transcriptome for inferring cell identity. And this is a joint work with Tianming Zhou and Jian Ma. A fundamental question of single cell analysis is what constitutes a cell's identity, and how do different factors like intrinsic type or spatial position interplay to give rise to observed gene expression. And a popular and appropriate method for modeling these different interplay of factors is matrix factorization. In the matrix factorization formulation, each factor corresponds to a metagene, which captures the expression profile of that factor. And then the mixing of these different metagenes gives rise to the observed gene expression. And this has been used in a number of methods. However, what is needed is a method that can capture the spatial patternings of the different metagenes if our interest is to really uncover the spatial factors. Importantly, if these latent mixings aren't independent, but probably exhibit some spatial coherence. And if we can model these relationships during inference, then we could learn more informative representations of a cell's identity. And recently, there's been a lot of work in spatial transcriptomics methods. And some of these methods are genome-wide, and even targeted methods can acquire now several thousand genes. So there's a need for such kinds of tools that can look at the spatial context of cells and disentangle the influence of different spatial relationships. And that's the motivation for the development of our work of SpiceMix, which is a general tool for analysis of spatial transcriptomic data that can be applied to any method. It works by extracting an expression matrix of the gene counts from the data, as well as a cell-to-cell graph. And given these two aspects of the data, it puts them into a novel probabilistic graphical model. And in this model, the observed expression follows the standard NMF formulation. But the key technical innovation is that the latent states or factors of the NMF formulation are related to one another through the graph. They're hidden states in a hidden Markov random field. And then the spatial potential of the hidden Markov random field captures the affinity for certain metagenes to be near each other or to repel each other in their spatial context. And an important note is that we don't assume a priori to know what the affinity for certain metagenes are, which would be the standard HMF formulation, but we learn that from the data, which allows the model to be more expressive. And then once we fit the model to the data, that provides us with the cell type composition X here on the right, which are the different latent representations of these cells, which we can then use to explore the different factors of the cell's identity. And to fit the data to the model, we treat it as a maximum a posteriori problem, where we're solving for the likelihood of the data given the parameters. And to solve this, we proposed an alternating map optimization method, which has some technical challenges since we're treating the hidden states as continuous variables and not discrete in a standard HMRF, like in a standard HMRF. And because we're trying to learn these spatial affinities from the data rather than assuming it. And if you're interested in the technical details of this, I encourage you to look at our paper, which will be available soon. To evaluate SpiceMix, we created a synthetic data set of the mouse cortex, which is a tissue of interest in many recent spatial transcriptomic studies. The cortex consists of several layers of tissue, and three major classes: excitatory and inhibitory neurons and glial cells. Some of these cells, like excitatory neurons, have layer-specific patterns, whereas others exhibit sparse or diffuse patterns through the tissue, like inhibitory neurons. So in our simulated data, underlying these major cell types are the metagene compositions that give rise to them. And we constructed the data such that each cell type has a common metagene that it shares, and then there are subtype-specific and layer-specific metagenes that define these different subtypes or spatial patterns. And then the observed expression just is related to those compositions by the standard NMF formulation, where we added some noise to test the robustness of the method to variation of noise. And we also tried different data sizes to characterize the method's robustness to that as a factor as well. What we found is that SpiceMix is able to recover these spatial patterns of layer-enriched metagenes much better than NMF can. On the right here, we're showing the expression patterns of several metagenes. And you can see that for metagenes 10 and 11, for example, SpiceMix is able to capture these layer-specific patterns, whereas NMF, its expression is diffuse across the entire field of view or even multiple layers. And quantitatively, we found on the bottom left here that SpiceMix is able to reduce the error of the estimates of both the hidden states and the metagenes across the board, not just for layer-specific metagenes, but for across cells throughout the sample, we find a pattern of SpiceMix reducing the relative error. And for we use different regularizations of the spatial information in SpiceMix, which are shown here, why there's two SpiceMix algorithms, to see how that affects the accuracy of the results, but found that SpiceMix is able to do better across the board. We then clustered the cells based on this latent representation and examined them in their spatial context. And what we find is that because NMF isn't able to capture these layer-specific metagenes, it struggles to find layer-specific cell types. In particular, the excitatory neurons, which are the green-colored cells here, it struggles with. And you can see the dark green, there's a dark green excitatory neuron in NMF that's found diffuse throughout all of the tissue, whereas in the simulation and in the SpiceMix result, those green cells show the layer-specific patterning that we expect of excitatory neurons. In comparison to HMRF, SpiceMix is also able to better find these sparser patterns of the inhibitory neurons or other cell types, whereas HMRF, because it assumes a spatial smoothing pattern of affinities and it can't learn different patterns, it produces an overly smooth result, and it even blurs across the layers of the different excitatory neurons. So then we sought to apply SpiceMix to real data of the mouse cortex, in this case by a recent method called STARmap. And in this data set, it consists of a single sample of 930 cells and just over 1,000 genes. We took SpiceMix, we applied it, we extracted the hidden states of the NMF HMRF model, which are then the latent representation, and then we did some analysis. And the first step was to apply clustering to the latent space to look at different patterns of cell types. And what we found was that SpiceMix was able to reveal several subtypes that weren't found in the NMF results. In particular, it found a rare SST inhibitory subtype and some oligodendrocyte and excitatory neuron subtypes. And I'll just mention that we use the CH index as a metric to decide the number of clusters for each method, so that it's an objective metric. And we also looked at known marker gene expression within these subtypes to confirm that these weren't spurious subtypes or noise, but are actually legitimate established subtypes. And if we look at the spatial patterning of these cells in situ, we can see that SpiceMix is able to recover the layer-specific patterning that we expect, particularly in the excitatory neurons, and was even able to improve over the original STARmap study. But we can also see that it can find different spatial patterns, not just layer-specific patterns like those neurons or these two oligodendrocyte types, but even this oligo2 type that's found diffuse throughout tissue. And again, if we were running HMRF on this, because it assumes a spatially smooth output, it wouldn't be able to find these different types of patterns, or SpiceMix can learn them from the data itself. And if we look at the latent metagene representation underlying these cell types, we can see that SpiceMix produces spatially coherent patterns. In this case, either layer-specific patterns or sparse patterns, which are probably reflecting inhibitory neurons. Whereas NMF, in contrast, shows diffuse patterns throughout the sample. Even for metagenes that seem to be layer-specific, there's still representation of that metagene in other regions of the tissue, which we expect is why SpiceMix is able to better refine these subtypes. And beyond just discrete cluster assignments, we wanted to know if SpiceMix can reveal insights into the identity of these cells by its latent factors that it learns. And one intriguing example that we found was among oligodendrocytes of these two subtypes. And we noted that they both express two metagenes, 12 and 13, at high levels, but at anti-correlated levels. And in fact, the cells seem to lie on a continuum between these two cell types rather than just belonging to discrete types. And we hypothesized that this could be the myelination process of oligodendrocytes. And we confirmed this by, or tested this by looking at Go terms that are related to myelin sheath formation and testing them against this proportion of these two metagenes, and found that a significant number of the genes in myelin sheath formation were significantly correlated to this seeming continuum between these two metagenes, which confirms that this could be what SpiceMix is revealing. And I'll also just note that SpiceMix can be applied to other methods, not just STARmap. We also ran a similar analysis on SeqFish Plus data and of another data set of the mouse cortex, and found a similar result where SpiceMix is able to reveal similar subtypes that can't be found by NMF, and again is able to find the layer-specific patterning of different cell types, like the excitatory types in the data. So in conclusion, we believe that SpiceMix is an effective tool for modeling the spatial transcriptome. We evaluated it on synthetic cortex data and showed its effectiveness, and we applied it to real-world data like STARmap and showed how it can refine cell type assignments and find finer subtypes, and also reveal interesting aspects of cell identity by the latent factors. And we believe that as spatial transcriptomic data becomes more widely available, SpiceMix will be an important tool for analyzing this data. And with that, I'd like to acknowledge co-authors Tianming Zhou and Jian Ma, and mention that our code is available on GitHub, and we'll have a bioRxiv preprint soon. And I thank you for your attention.
All right, so now let's look at the questions. Yeah, so the first question is from Anshu. Is SpiceMix able to integrate spatial transcriptomic data sets with complementary non-spatial single-cell RNA-seq data?
Yeah, that's a great question. Yeah, it is able to, and that's something that we're intending to do in our follow-up work. There's some consideration that has to be made to normalize the two, such that the characteristics are the same for the single-cell RNA-seq and whatever transcriptomic spatial method you're using. But yeah, that's I think a really important direction to help refine inference.
Okay, thanks, Ben. So I think people are still typing in questions. So maybe I can ask you a question now. So I think it's a super interesting talk, and it's a super interesting problem to consider the spatial location of single cells and the gene expression data when we talk about cell types. My question is a bit more general. So in general, do we have any knowledge about, whether we consider that cells which are located close to each other, whether they are more likely to be the same cell type? It's probably like more conceptual question.
Yeah, that's the assumption that the standard is in a standard HMRF. Like when you're using it for image segmentation, for example, you assume that pixels next to each other are of the same type. It depends on the cell type. Like the excitatory neurons, which are densely populated, do tend to be near each other, but inhibitory neurons are very sparsely populated through the tissue, so they probably won't be next to each other. They'll be on their own. So that's part of why we created the method to learn the spatial affinities, because it depends on the cell type, if that is true or not.
Okay, thanks, Ben. I think we actually can move to the next talk. So if audience has any more questions for Ben, please contact him or join the Cafe Connect platform.